Good morning, everyone. My name is Karen King. I am the Community Affairs Liaison for the Office of New Haven Affairs. Thank you for joining us for our October 2023 Community Breakfast. We hold these breakfasts on the first Thursday of every month during the academic year. And you will get notifications if you're on our mailing list, our email distribution list, about the each upcoming month's topic and our list of speakers. I have a few announcements before we get started. First, today at 4 p.m., the Yale Office of Sustainability and the Yale Program on Climate Change Communication will be hosting a walking tour of Yale's campus to learn about how the university is working to innovate climate solutions through both research and campus operations. The tour starts today at 4 p.m. It kicks off at Phelps Gate on College Street, and you'll learn a lot of things in the hour-long tour, from buildings with next-generation clean energy systems and bird-safe window treatments to green spaces that serve as community gathering spaces and promote biodiversity. I will put a link in the chat to, and I invite you all to join the tour. You're going to meet a couple of great people who work here on campus, and you'll learn a lot about Yale's climate change and sustainability efforts. Next, one of my favorite events, actually there are a lot of great events that are hosted by the Shops at Yale downtown, but one of my favorite events, as you can see from my poster behind me, is the annual Chalk Art Festival. So this year's festival will take place on Saturday, October 21st, if the rain holds off. If it doesn't hold off, it will take place on the 22nd. And it takes place from noon to 4. There will be 2 locations where these amazing chalk art artists will be drawing. One is outside of the Yale University Art Gallery, which is pictured here, and the other is on Broadway Island, which is across from J. Crew. It's a family-friendly event. There are a ton of great activities that go on besides watching these amazing artists create these works of art. First 200 kids to arrive will get some free chalk so that they can make their own creations in a family fun zone. There's a magician, face painting, live music. It's a really great fun day. So hoping that the rain holds off. If it doesn't, please join on Sunday, same time. But keep your fingers crossed for a nice sunny day on Saturday. And lastly, I have talked often about Yale's New Haven Hiring Initiative, which is a wonderful initiative at the university. There's a strong team that is constantly out in the community, raising awareness about positions that are open at the university and the process for hiring. They have a partnership with the Q House, where they host employment readiness sessions. They also host office hours. There is a great, they have great sessions every other Monday, where they bring in local employers to talk about their frequently filled jobs and how to engage in their application processes. They also provide workforce development resources to local residents as well. There is a great calendar of events that are coming up. For instance, on October 16th, the Workforce Alliance will be presenting about opportunities at the American Job Centers. On November 6th, the Construction Workforce Initiative is a local organization that trains people who want to get into the construction business and as workers. So it's a great training program. They also have OSHA certification classes as well. So that's on November 6th. November 20th, Yale Public Safety and New Haven Works will be co-presenting about opportunities and application processes there. Yale Public Safety has a number of positions that are available. They're going to be talking about that on November 20th. On December 4th, the Yale IT department will talk about early careers at Yale. And on December 18th, Marrakesh will be a local nonprofit will be talking about their organization and their Academy for Human Service training. I am going to put this information in the chat, and Crystal, I'll also send it to you. Thank you for that message as well. But there's a sign-up sheet, and the calendar of events will be on the sheet, so people are encouraged to come on, sign up. If there's a wait list, please join the wait list. I'm pretty sure almost always there is space for everyone, even for the popular programming. So that is it. If you have any questions, let me know. Again, I'll put all that information in the chat. So on to this morning's speakers. We have 4 great speakers this morning. First is Jordan Cardenas. Jordan is a 5th-year PhD student in Yale Immunobiology, studying strategies for overcoming resistance to immunotherapy and lung cancer. He is happy to share his experience with cancer therapies and will be happy to answer any questions. Alex Dalrymple is a senior at Yale College, studying molecular biology. She conducts research on immune tolerance in the Yale Immunobiology department and is looking forward to speaking about the biology behind immunotherapies. Jenny Liu is a 2nd-year Master's in Public Health student in the Yale Biostatistics department. She's working on potential immunology therapy on a secondary brain tumor. She is excited to share her knowledge and engage in conversation about cancer immunotherapy. And Kavita Israni-Winger recently completed her PhD in immunology and is now a 1st-year medical student at Yale. She worked on inflammation and metabolism and looks forward to discussing science with everyone. Please help me welcome all of our wonderful speakers this morning, and I'll hand it off to Kavita. Welcome, everyone.
Hi, everyone. Thank you for being here today. We're really excited to talk to you guys. I'm going to share my screen to start off the presentation, but it looks like it's not letting me share my screen here. Karen, it looks like it's not letting me share my screen.
Do you want to send your presentation to someone else, or?
I can't, it's not my computer, it's because of the participant screen sharing.
You know what? It's not a you problem, it's a me problem.
No.
Do it now. My apologies.
No problem.
You should be all set now.
Okay, yep. Alrighty. Start the slideshow. Okay, so I'm not going to spend too much time talking now, but these are pictures of everyone who's presenting today. And before we start, I wanted to let Jenny Liu give a little bit of a personal story about her experience with her family and cancer immunotherapy, and then we can get started on the actual biology. So Jenny, go ahead.
Hi, my name is Jenny. So I would like to share my family members' experience with cancer immunotherapy treatments. So my aunt Bing had a colon cancer, but then transferred to liver. So she was treated with immunocheckpoint inhibitors, and within 5 months, the tumor in the colon was completely gone. Well, however, in the liver, it cannot be treated effectively with immunocheckpoint inhibitors. So currently, she's undergoing traditional chemotherapy and surgical resection. So I just wanted to say, with all of those cancer treatments methods and with the combination of those methods, people can really benefit from it and get cured. Thank you.
Thanks, Jenny, for sharing your story. I know that a lot of us have had some interaction with cancer at some point in our lives, I'm sure. And so today we're hoping to give you a little bit more insight on what some of those cancer immunotherapies look like and the importance of doing that kind of research so we can do more and have more success stories like Jenny's on. So I will so this is going to be a general overview of what we're going to talk about today. We'll briefly talk about the history of cancer immunotherapy, then the biology of immune cells and cancer, and then Jenny will tell us a little bit about current immunotherapies, and then I will talk a little bit about current research and future immunotherapies. I just wanted to put out there that I have to run at like 8:55, so if I look like I'm scrambling and running out of here, I apologize, but I really am excited today. So Jordan's going to start us off with cancer immunotherapy.
Yeah, thank you, Kavita. Hi, my name is Jordan. And yeah, I wanted to talk a little bit about the history of cancer immunotherapy. I think these types of things are important to realize, where the ideas came from and sort of how we came to understand really the immune system and cancer a lot better because of immunotherapies, even if we didn't think of them as immunotherapies at the time. So that starts really way back in the 1890s and early 1900s with this guy named William Coley. He was a surgeon that specialized in sarcomas, which was a type of bone cancer. And so he treated a lot of different patients with sarcomas, and he kept extensive records on their medical history of all these patients. And so he would often go back to these records and search through the records and to try and learn about the patient and the development of their cancer and how he could best treat them. In one of his patients, he did see that the tumor completely regressed, so completely went away, only after the patient became infected with this erysipelas, which was a type of bacterial skin infection. And you can go ahead and I think you might have missed something, but oh, never mind. Sorry about that. So because of that idea, he came up with the idea of what we now call Coley's toxins, starting in about 1893. The idea was to take the bacteria that causes the skin infection, Streptococcus pyogenes, and kill it with heat and inject that into patients that had sarcomas. And so he began treating these patients with the heat-killed bacteria, and he tested a lot of different formulations and mixtures of the bacteria. Sometimes the mixtures led to no effect on the tumors. Sometimes the mixtures led to patient mortality because of additional bacterial infections they got. But after some time between the 1890s and 1936, he settled on a mix of Streptococcus pyogenes and another bacteria, Serratia marcescens, that really seemed to work in patients. And all in all, throughout that time, he treated, it's estimated he treated about 1,000 patients with sarcomas, and he saw regression of the tumors in about half of those. So he didn't necessarily know what was causing the tumor regression and how the mechanism of how that was working. But in effect, the main point here is that he saw that a bacterial infection ultimately led to tumor elimination. And so we didn't have a great understanding of the immune system at that point. But as we would learn more, we would learn more about how the immune system gets engaged during bacterial infection and how that can eliminate tumors. So if it works so well, why stop using Coley's toxins? There are a couple of reasons for that. Coley was fairly meticulous about his formulation, but it wasn't, especially at the time, it wasn't always easy for other surgeons to follow this and replicate it exactly. So they had some of those variations and effects that Coley saw in the beginning of his work. And those variations in the formulation, as I had mentioned before, could lead to wild variations in the efficacy of the treatment, and in some cases could end up being fatal for the patients. Another reason that Coley's toxins weren't really widely used at the time and going forward has a little bit to do with sort of the politics of science. There was another researcher at the time named James Ewing. He was the founder of the American Cancer Society and the American Association for Cancer Research. So he was fairly well regarded within the field, and he just disagreed with William Coley's methods and didn't think that it was efficacious. He actually pioneered radiation therapy, and so because of his influence, the sort of standard of care became radiation therapy, and people kind of stopped using William Coley's methods. But as I mentioned, as we came to understand more about how Coley's toxins stimulated the immune system, retroactively, we think of Coley as the father of immunotherapy. So next, I want to jump forward a little bit and think about the concept of tumor immunity, or how the body can recognize tumors in a specific way and memorize that recognition. So these experiments started back in about 1940s, 1943 or so, looking at tumors in mice. And some researchers found that when you inject a large number of tumor cells into a mouse, the mouse begins to grow a tumor, and then after some time it gets too big and the mouse dies. However, When they injected a smaller tumor than the larger tumor, they noticed that the mice began to develop a tumor, but then after some time, that tumor went away. And so without any treatment or intervention. So this mouse that was given less of a tumor, the tumors went away and the mouse was able to survive. They then took this a step further, and in those mice where the tumors went away, they injected another of the same tumor and found that the mice didn't develop a tumor at all, and continued to live healthy lives. So the main point of this experiment was to see that mice can develop specific immunity to tumors, and that's something going on within their own body and not with any sort of treatment intervention. So similar experiments were performed between the 1940s and 2000, but the next big leap was made in 2001, where a different research group took normal mice and mice that are deficient for certain types of immune cells and treated them with the chemical carcinogen. So this graph shows the results. MCA is the carcinogen and WT or wild type are the normal mice. RAG2 mice, as I mentioned, lack a certain type of immune cell, and you can see that over time on that MCA treatment, the mice without that specific type of immune cell develop a lot more sarcomas than the normal mice. Again, they took it a step further and looked at different genes that control development of different types of immune cells that Alex will get into in a little bit. But again, you see, compared to the normal mice, all of those mice with immune cell knockouts develop more tumors naturally. And so this really showed that immunodeficient mice develop tumors at a higher rate. Again, suggesting that the immune system has something to do with tumor control. This didn't only happen in mice. This was also seen in humans, and this is a little bit more recently tracking this type of thing. But in humans, there is a national registry of patients that have immunodeficiency genetic disorders. And so those patients are marked with the red bars and the purple bars here compared to patients that are not on this registry in the blue bars and green bars. And you can see at any age, there are more patients that have on that immunodeficiency registry that have developed tumors than patients that are not on that immunodeficiency registry. So patients with immunodeficiencies develop tumors at a higher rate and are at higher risk for developing cancer. I'll jump back a little bit because, as I mentioned, some of these therapies were developed without a full understanding of how they worked and what they did. But the first FDA-approved immunotherapy came in 1972. Between 1972 and about 1992. This was actually from a group here at the Yale School of Medicine who discovered this back in 1972, this lymphocyte activating factor, which is a molecule that activates immune cells. A little bit later, this molecule was renamed interleukin-2 or IL-2, and it was purified so that it could be administered to patients therapeutically. And researchers found that therapeutic administration of the IL-2 during clinical trials in cancer patients, they were initiated and seen that it can lower the tumor progression of kidney cancer and then later melanoma. So in 1992, IL-2 became FDA approved, and so that's why it's the first FDA approved immunotherapy. So just putting all of that together, what I've been talking about, going back all the way back to Coley and back to what we've talked about right now, what we've learned since Coley is that, as I mentioned, a bacterial infection does cause some sort of stimulation of the immune cells, and it turns out it will cause the activation of what are called innate immune cells that Alex will talk about in a second. But those innate immune cells use IL-2 and other such molecules to activate adaptive immune cells. And we've seen that these adaptive immune cells are really what's responsible for going into tumors and killing the tumor cells. The next. Thank you. Yeah. So with that, I'll toss it over to Alex and talk a little bit more specifically about the immune cells.
Hi, everyone. My name is Alex, and it's a pleasure to be here. It's a joy to talk with you guys about the biology behind the immune cells in cancer. So for my section, I'll first give you a brief overview of the immune system, where we'll focus specifically on the different types of cells that are used in current immunotherapies. We'll then dive into the immunotherapies, and we will look into the first type that I'll talk about, which are immune checkpoint inhibitors. After we talk about immune checkpoint inhibitors, we'll talk about one other really pertinent form of immunotherapy that's currently used clinically, and that's CAR T-cell therapy. So you might have heard of immune checkpoint inhibitors and CAR T-cell therapy. And my goal is, after talking with you today, you understand a little bit more about the biology behind these different mechanisms, and then I'll pass it over to Jenny, who will talk about other kind of new and upcoming forms of immunotherapy. But to begin, we'll talk about the immune system. And so there are two main branches of the immune system that are worth mentioning. And in general, so in the immune system, you have what's known as the adaptive immune system. And as Jordan was talking about, the adaptive immune system can be activated by this innate immune system. It gives you a slower and more specific response. Another feature of the adaptive immune system is that it can remember the germ. So what's really neat about the adaptive immune system is where the innate immune system, which I'll speak about in a second, works really rapidly, and it's your front line of defense. After its activation, you get the slower and more specific response with the adaptive immune system. So you can imagine that if it's slower and more specific and can also remember the germ, this would be a really helpful branch of the immune system to look into for immunotherapies. And so this is specifically the branch of the immune system that we'll discuss today. But to give you a little bit of a background on the steps that take place before activation of this adaptive immune system, you also have the innate immune system. So the innate immune system is what is activated as soon as you get infected with something. For instance, if a bacteria is found in a body tissue, this is a non-specific response. So it's just detecting broad patterns that are found on different types of common pathogens like bacteria and viruses. And so with this non-specific response, you also have really fast response. And so what typically happens is if the innate immune system can't contain something like an infection, or if there's a cancer cell that's expressing a certain protein, the innate immune system likely won't respond to that. And if it doesn't respond in this quick time frame, then the adaptive immune system is activated, as Jordan kind of alluded to. We'll dive a little bit more into the adaptive immune system, since that's what's really pertinent to current immunotherapies. So the innate immune system is very interesting in and of itself. So in the adaptive immune system, we have two cell types that I'll mention today. And those are these B cells or B lymphocytes, as well as T cells. So on the next slide, I'll discuss a little bit of the difference between these two forms of cells. So lymphocytes, again, we have T cells and B cells. T cells are what we'll predominantly be focusing on, at least in my section of the presentation. And T cells are really neat because they develop in the bone marrow, but then travel to the thymus, which is a little butterfly-shaped gland kind of by your neck. And they mature in the thymus. There are 3 different types of T cells. We're going to focus on one specifically, which is known as the cytotoxic T cell or the killer T cell. And you can imagine, and we'll discuss about the biology behind it, but if it's a killer T cell, that's probably very helpful to target and kill cancerous cells, which is currently how these cells are being used and manipulated by clinical immunotherapy. Just for some background on B cells as well. B cells mature in the bone marrow, so they develop in the bone marrow and stay in the bone marrow for maturation. They're really neat because they can kind of go into 2 paths. They can become these plasma cells. These plasma cells produce antibodies. So antibodies are really helpful in a lot of different ways, but they can neutralize, for example, in this picture, neutralize viruses, do lots of other different things, but they also have specific response, which makes them a part of the adaptive immune system and can also be looked at as a form of therapy as well. They're also memory cells, so this is where your memory comes in, and they can produce antibodies after the second exposure or future exposures with a particular pathogen. But in this presentation, we'll really be focusing on this guy. So this is your cytotoxic T cell, your killer T cell. And throughout the presentation, I think it's helpful to remember when we're talking about these cells, this little guy, so he has an arrow and a bow, and he specifically targets a cell that has a certain flag, essentially, that he can recognize with this little receptor on his head. So we'll talk about that a little bit more. But again, these are the cells that we'll really be focusing on this presentation. They produce these toxic agents that can kill our target cells. And so diving into these cytotoxic T cells, this is a kind of complicated looking figure, but what I like to remember is the way these cells are activated and know what to kill precisely is almost like a lock and key mechanism. So I always like to think about what's going on here is if you want to specifically kill a cell, you do want this to be specific. You don't want autoimmune disease, which can happen when these cells are activated against body tissue. And so this cell on the bottom is a body cell. And so, yeah, I like to think of this holding a little peptide cargo, which is found from the cell, as a key. And this key fits in a specific lock on a T cell. And these T cells are in your body. And if they recognize the specific lock, they'll become activated and can attack this cell. So this is essentially a signal that tells the cell that it needs to be targeted by the cytotoxic T cell and killed. And so this little MHC complex, that's found on body cells, and cancer cells are body cells, so it's also found on cancer cells. And it displays protein fragments from the cell to the T cell. That makes the specific shape of the key that fits into the T cell's lock, which is its T cell receptor. So the T cell receptor lock is found on these cytotoxic T cells and matches a specific MHC encoding. So this just gives your specificity, and you'll see later in the presentation why this is so helpful and how it's being used in research to treat patients clinically. So that's a little bit of an overview on antigen presentation is what this process is called. We'll dive into this little magenta colored receptor, which is an immune checkpoint. And the immune checkpoint, I like to think of it as like a brake. So if we have a cell that has the right key and the lock of the T cell fits it, but we still don't want to target this cell, it's a normal body cell and it's healthy, we can have this braking system. And so if this immune checkpoint binds to its binding partner on a cell, even if it has the right lock and key, the T cell won't kill this cell. And so it's a big stop signal. The cell can continue to survive. And so looking at that kind of in more of a schematic view, we have just normal body tissue. And if our body tissue matches there we go, if it matches the particular lock and key again, but you have this binding partner, these cells will survive. So these cells will be fine. The signal says, stop, I'm healthy. It's a braking system. And this helps prevent against autoimmune disease. However, in cancer, we want to break these checkpoints. We don't really want cancer cells to express this little receptor that says, stop, I'm healthy. And so sometimes in cancer cells, they don't express this receptor. So this is going to be found in probably a lot of the patients that Jordan was talking about when they were given Coley's toxins, they had remission and their cancer was cleared. A lot of these cancer cells likely didn't express this break, and so they could be killed. However, what can sometimes happen in cancer cells is if we kill these cells that don't have the break, left is these ones that are expressing this binding partner break. And so this immune checkpoint, which I'll call PD-1, you might have heard of PD-1 therapy. We'll talk about that in just a second, or PD-L1 therapy as well. But in this case, this cancer cell has the right locking key, so it could be recognized by the cytotoxic T cell, but is not because it's expressing this break. And so this process, again, that would be a stop signal. So the cell would survive, the body would think it's healthy and it's not. But clinically, what if we think breaks, or what if the breaks are blocked? And so this is the whole purpose of immune checkpoint inhibitors. They bind to either the immune checkpoint on the T cells or its ligands, it's the receptor, or its ligand on cancer cells. And so, for example, you can have both anti-PD-1 therapy, which binds to the receptor on the T cell, so that binds to the T cells break, or it can bind to the cancer cells break, which is anti-PD-L1 therapy. And so blocking either of these signals will allow your T cell, if it has the right lock and key with another cell, to kill that cell, regardless of if it's expressing these breaking mechanisms. So that's a little bit of the biology behind immunotherapy, or immune checkpoint inhibitors, rather, as a common form of immunotherapy. And the key takeaway from this slide really is that there are these forms of immune checkpoint inhibitors, but the different types of cancers express the immune checkpoint inhibitors differently. And so you can see that in each category, we have different forms of cancer that are currently approved in their treatment with different forms of immune checkpoint inhibitors. And so in the clinic, typically, testing is performed to see the levels of these proteins before treatment, which allows for a diagnostician or an oncologist to provide the right treatment paradigm for patients depending on what their cancers might respond to. So this is a way to get more personalized treatment by looking at what each cancer expresses. And so that's the main idea behind immune checkpoint inhibitors. They're really helpful. A lot of patients, like Jenny, her family has been touched by cancer and was really impacted by immune checkpoint inhibitors. So these are really remarkable form of treatment that's widely used clinically. And now we'll move on to CAR T-cell therapy as the last form of current therapy that I'll discuss today as being really pertinent in the field. And where we talked before about this immune checkpoint, this PD-1 right here, here we'll talk about the T-cell receptor. So the T-cell receptor, that is essentially looking at the lock of the lock and key kind of way of thinking about things. And so in CAR T-cell therapy, CAR stands for chimeric antigen receptor, which is just a little bit of jargon to say that T cells are engineered to make these T cell receptors that are specific, and they're specific to a protein produced by cancer cells. So this is, again, using this ability of the adaptive immune system to be specific and manipulating that in such a way that we can treat individual patients' cancers. It's a very personalized form of medicine. And to talk a little bit about how we can make these this CAR T-cell therapy, here we see we have a patient, and with our patient, you can collect the blood. So clinicians will collect the blood of the patients, isolate their T cells that are from this patient. They'll then reprogram these T cells to make them express this particular key. And so the shape of the key is dependent on whatever type of, the shape of the lock is dependent on whatever key the cancer cell has. So you'll reprogram these T cells, multiply them outside of the patient's body, and then give them right back to the same patient. And so these CAR T cells will then attack the cancer cells that they were engineered against. And so this is a really neat form of therapy. And to kind of paint a picture, make this feel really personal, I wanted to share the story of the first patient who received CAR T cell therapy. Her name was Emily Whitehead. This is Emily. And she was diagnosed with a form of leukemia. I think I paused the recording. There we go. And so this is Emily, and she's currently 12 years out, a really big advocate for CAR T-cell therapy and for its research. So that's her and a little bit about her story. And she really pushed this field forward. And just looking finally at the types of cancers that are currently treated by CAR T-cell therapy, the main takeaway of this is that these forms of cancer are blood-based cancers. And so CAR T-cell therapy, just because it's hard to get your blood into solid tumors, is currently being used in different forms of blood-based cancers. However, there is currently a lot of research going on all around the world to apply this to solid tumors. So I will pass it over to Jenny to talk about current immunotherapies.
So hi, this is Jenny. So I'm going to talk about the current immunotherapies. So with all the good things, amazing treatment of immunotherapy, however, nothing's really perfect. So I'm going to talk about the effectiveness and also side effects of this immunotherapy, namely checkpoint inhibitors and CAR T-cell. Okay, so I will start with the checkpoint therapies, as shown in the graph, as Alice talked about, a brief review of what is immune checkpoint inhibitors. And the most common treatment for this method is using anti-PD-1 or anti-PD-L1 inhibitors and or a combination of CTLA-4 inhibitors. So here are some examples of the drug that is, could you go to next slide? Yeah, so here example of the drug is pembrolizumab, nivolumab, and cemiplimab. So the effectiveness of the drug really depends on several factors, including type, stages of the cancer, patient's overall health, and individual responses to the treatment. But in general, tumor shrinkage will occur within weeks or several months of the starting treatments. And the good news is that it shows an advantage in controlling tumors compared with other conventional therapeutics and has fewer side effects. So for example, as Alex talked about, checkpoint therapy shows novel responses in treating skin cancer. So studies have shown that the long-term overall survival rate could be as high as 44%. And as shown in this diagram, with 1 to 3 months of initiation of the treatments, the patient shows responses and it has disease-free progression time as long as 15 months, while chemotherapy only have weeks or months disease-free progression time. So this is another graph from one of the scientific paper showing the elimination of skin cancer following 3 months of using checkpoint inhibitors. So here they use the combination of anti-PD-1 and CTLA inhibitors. So checkpoint does have side effects. This is because it does not necessarily kill cancer cells directly. Instead, it enable immune cells to recognize and attack cancer cells more effectively. So because immunotherapy drugs unleash immune cells, inflammations may occur in organs such as colon, causing diarrhea, in lungs, causing coughing or shortness of the breath, in skin causing rash, and in liver causing alleviation of liver enzyme in the blood, and maybe other areas of the body. However, the good news is that it has fewer adverse events than the conventional therapies, and it only happens about 20% of the people given PD-1 or anti-PD-1 drug inhibitors, and occurs 40 to 60% with the combination of PD-1 inhibitors and CTLA-4 inhibitors. So if you had immunotherapy at any time in the past, report any new symptoms to a treating oncologist before self-medicating with the drug purchased over the counter. So for example, if you have diarrhea, taking loperamide might arrest the symptoms, but it won't address the root cause, which is inflammation in the large intestine. So uncontrolled inflammation of the intestine may lead to rupture of the intestinal wall, which can be life-threatening. So please always consult your oncologist before self-medicating. So second, I would like to share the effectiveness and the side effect of CAR T-cell therapy. So this type of treatment can be very helpful in treating some type of cancer, even when other treatments are no longer working. So in one of the study, 9 out of 10 people with acute lymphoblastic leukemia whose cancer did not respond to other treatments or have full remission with CAR T-cell therapy. So remission means that the cancer that can be detected in this test. So in a study, post 6 months of CAR T-cell therapy, 76% of the patient achieve remission, and only 33% of them experience immune effector cell-associated neurotoxicity syndrome, which is a common side effect of CAR T-cell therapy. So patients treated with CAR T-cell therapy don't experience the hair loss that typically associated with chemotherapy. They are, however, at the risk of severe but short-term and manageable side effect called cytokine release syndrome, also called CRS. So cytokines are proteins that immune cells release when they attack an infection. And CRS causes a high fever, severe flu-like syndrome. So for CAR T-cell patients, fever is actually a good sign that therapy is killing tumors. So CRS can be serious but manageable in hospital. So C-cell can be serious but manageable in hospital. A drug called tocilizumab can be entirely reverse CRS reasonably quickly. An additional potential side effect could be in some people include neurologic symptoms such as confusion, trouble speaking. So these short-term effects are generally fully reversible. The long-term effect of CAR T cell therapy may not be fully known since the therapy has only available for 1 decade. But we do have 10-year experience that had not had any unexpected long-term complications. And we will continue to observe and report openly to any new findings of concerns. So thank you for listening. This is my part. Next, Kavita will talk about the future direction for cancer immunotherapy.
Thank you so much, Jenny. That was really good. Okay, so today I'm going to focus on two little things, and I'm going to try not to breeze too quickly through them, but at the end, Jordan, Alex, and Jenny all have a lot of expertise in this area and can help answer questions where I can't. So today I'm going to talk about cancer vaccines first, and then bispecific antibodies. So the whole idea behind cancer vaccines is that it's supposed to kickstart the immune system in a very specific way. So one issue that we have with a lot of cancers is it's not always an immune response to treat with checkpoint therapy. So what the vaccines are intended to do is to kickstart the immune system when it's not doing it for itself. There's a couple different ways that this can be done. One of them is by using a predefined antigen. So that's if we already know within a cancer that there are specific proteins or molecules associated with that cancer that the body can recognize and then attack. In those cases, what we can do is we can take a protein that lots of people with that same type of cancer have, that's the shared in the blue here, and we can teach the immune system to attack anything expressing that. Or we can take a more personalized approach and we can take a protein that we know that a specific person's cancer is expressing and teach the immune system to attack that. Another way to do it, and this is incredibly important, is by an anonymous approach. And this is when we don't understand what makes the cancer unique and what to teach the body to attack when it's supposed to come after our cancer. So to do this, you actually take up parts of the cancer, of the tumor itself from the patient, you ground it up, and then you feed it to the patient's immune system and teach the patient's immune system what to attack and let the immune system figure it out. We don't know what the antigen is, but the immune system might know. So that's a brief background about the different types of cancer vaccines and how we can make them. One successful one is this one called Sipuleucel T, which is a vaccine that treats prostate cancer. One nice thing that we know about prostate cancer is that there is a prostate-associated protein. So I don't actually know that that's what it stands for, but it's called PAP, and it's a protein that prostate tumor cells express that are unique to prostate tumor cells. What we can do to help kickstart the immune system in patients that have this cancer is we can take the cancer or protein, so that antigen that Alex had talked about before, and then we feed it to a specific type of cell in our immune system called a dendritic cell. The dendritic cell is then activated and then teaches the T cells, those killer T cells that attack the tumor, who they need to attack. And then ultimately you can get killing of the cancer. I'm showing some data here for you. There's two graphs that show similar things. I want you to focus on the big one here. You can see on the y-axis, we have percent survival of patients, and on the x-axis, you have survival time. So essentially how long that they can survive with this diagnosis of prostate cancer. Sipuleucel T, so people treated with that prostate cancer vaccine, are in red, and you can see that they are able to survive, a higher percentage of those patients are able to survive longer after their diagnosis of prostate cancer compared to those who were on a placebo, so who are not receiving that vaccine. Next, I'm going to talk about bispecific antibodies, which are another very cool engineering feat that has been used by the immunology community to try and target cancers more specifically. So earlier, Alex talked very briefly at the beginning about antibodies, which are produced by B cells, which are a cell in our immune system. Antibodies come in very many different types and flavors, but typically one antibody will recognize one thing, and both arms at the top of this little Y here will recognize the same thing. So it's the arms that are responsible for recognizing things, and both arms recognize the same thing. So this purple antibody will recognize, say, a purple protein, and this green and yellow one will recognize a green protein. So they won't recognize the same thing. However, what immunologists have done to try and harness the specificity of the immune system is take one arm from one antibody and another arm from another antibody. So now you have this structure that can recognize two separate things. What that does is it can either bring two things together, it can activate one thing close to another thing, it can activate both things. The unique thing about it is that antibodies have a lot of different functions, and depending on how we engineer them, we can get very specific activities out of these products. So one specific antibody that has been used commonly and was the first one to be created is a bispecific antibody that can bring a killer T cell, in this case it's the green one called an effector cell, and a cancer cell close together. In doing so, it can also activate the T cell. So now the T cell can attack the cancer cell specifically because they're close together, it knows who to attack, and it's received an activation signal from being bound by the antibody. This can then ultimately lead to cancer cell death. One specific example of this is a therapy called Blincyto. Blincyto is used for treating B-cell lymphoma. And what it does is it has one arm of the antibody in blue here that can recognize a receptor on T cells, and then one in pink that can recognize receptors on cancer cells. It does that same thing that I talked about, where it brings them close proximity, tells the T cells go, so then the T cell can now kill this cancer cell. And it's a really effective way of treating this cancer. This is my demonstration of that here. This graph is a little less colorful than the previous one I showed you, but again, I'd like you to focus on the big graph here. On the y-axis, again, we have survival, and on the x-axis, we have the time since patients started being treated. So standard treatment right now for leukemia or lymphoma is chemotherapy. So they took patients that were receiving chemotherapy, and then they also put some patients on blinatumomab, which is the bispecific antibody or BiTE that I showed you earlier. And what that's capable of doing is extending the survival of patients for longer than if they had been on chemotherapy. So you can see that there's a higher probability of patient survival if they're being treated with this new cancer immunotherapy as opposed to classical or chemotherapy. Another thing that antibodies can be engineered to do is to carry important activating signals. So another thing that we can do to change the function of an antibody is by chopping off one of its arms and replacing it with a molecule that can activate and stimulate the T cell really strongly. This is a nice way to wrap it up because the one that's being used in clinical trials right now is an antibody with an arm that recognizes PD-1 on T cells. So that's that important immune checkpoint molecule that Alex told us about earlier. And then also IL-2, which is that really stimulating cytokine that Jordan told us at the very beginning, that alone is not as effective at treating cancer, or has a lot of side effects. Part of the reason IL-2 was an issue is because it activates immune cells everywhere, but what this antibody can do is choose the T cell that it wants to activate. So this therapy is called PD-1 IL-2V. And what they did, and this I want to point out, this graph here was done in mice, not in humans, because it hasn't reached that stage yet. But what they found is in the green line here, if mice with cancer were untreated, they passed away pretty early on. If they were given anti-PD-L1 therapy, which is a checkpoint therapy talked about earlier, they make it to about 12 weeks. If you give them PD-1 IL-2V, which is this new antibody I told you about, they do pretty well. But if you combine it with blue and with the checkpoint therapy anti-PD-L1 and with the new therapy for antibody PD-1 IL-2V, they do extremely well. You can see, again, a good demonstration, like Jenny told us at the beginning, of combining cancer immunotherapies to get really good outcomes from patients. So I really enjoy this way of wrapping up the talk because I think it demonstrates the power of immune checkpoint therapy that was talked about by Alex, the power of IL-2 and cytokines that were talked about by Jordan, and then also the power of combining all of those treatments, which were mentioned by Jenny at the very beginning in regards to her own family member. Okay, that is the end of the talk. We are happy to take questions. I have to run to a class now, so I'm actually gonna bump out and leave it to the rest of my colleagues to answer questions. But I really enjoyed talking to all of you today, and I'll see you next semester.
Thank you, Kavita. I certainly learned a lot today. I felt like I was back in college. A lot of great information. There is a question to start us off from Colette. She asks if one has had an autoimmune disorder like aplastic anemia that was cured 10 years ago, then might immunotherapy trigger the aplastic anemia to return?
I think maybe for that question, just to kick us off, and Jordan, Jenny, you're welcome to jump in. But I believe it depends on what type of autoimmune disorder was causing the aplastic anemia. So immunotherapy will, everybody kind of says it'll rev up the immune system. So kind of make your immune system more active broadly. And that's what Jenny was saying. There are a lot of side effects because you're not specifically killing what you want to kill. You're almost revving up the immune system. And so depending on what was causing the aplastic anemia, if you have immunotherapy and you reactivate a certain type of cell that was causing the destruction of the bone marrow, which is what happens there, then it's possible. But I think it just depends on kind of underlying what was causing that particular condition.
Thank you.
Jordan, did you have an idea?
Yeah, I'll add. I think that's correct. I think it's also, when your oncologist is going to prescribe the immunotherapy or whatever other therapy they might use, they'll take those types of things into account. So it might be, depending on the root cause of the autoimmune disorder, it might be sort of a combination of the of whatever drugs were used for that to kind of suppress the immune system, or they might just go away from immunotherapy altogether in favor of a different type of therapy.
Thank you.
Thank you.
Thank you. And thanks for sharing your cat with us, Colette. See him walking around.
Thank you. Thank you. Oh, yeah, sure.
Keith has a question. How do the T cells go about killing the cancer cell?
Yeah, I can take that one if you. Yeah, so the T cell, many immune cells produce certain molecules that sort of target cells and allow them to be killed. And I guess if we think, cancer is sort of a little bit of a newer disease in the sense that as we've lived longer, people develop cancers more because they're sort of your own cells kind of going rogue and growing uncontrollably. And that doesn't really happen when people didn't live quite as long. But if you think about it in that sense, T cells, their main goal from like evolutionarily from the beginning was to target our own cells that are not functioning correctly in some way. And so the way it does that is that our own cells have a lot of mechanisms of essentially killing themselves when they're not performing correctly, or when they're not functioning correctly, or when they're not performing their function correctly. And so the molecules that the T cells produce, examples include interferon gamma. Those cause those internal mechanisms within the cells to be activated to undergo cell death. Another example is perforin. That's a molecule that T cells will often produce, and that sort of embeds itself in the cell surface of cells and causes holes for all the internal components to sort of leak out, and the cell dies that way. So there's a couple of different ways, but those are the main two.
Great, thank you. We have time for one more question, if anyone has one. And just check the chat briefly. And Helen, I saw your question about the tour. I reached out, and I'll get back to you to let you know. Well, I don't think we have any other questions. I think people are still processing all the information that you all shared with us this morning. So I want to thank you for your time. I know you all volunteered to join us this morning, and I appreciate the effort that you put into your presentation and sharing, certainly waking my brain up this morning and sharing a lot of great information. So thank you so much for joining us. And thank you all for attending this morning. I hope that we will see you next month, where the Yale Prison Education Initiative will be presenting about the opportunities that they bring to incarcerated people who are incarcerated in Connecticut prisons. So please join us next month. Stay tuned. And again, Jenny, Jordan, and Alex, thank you so much for joining us. Bye-bye.